going to introduce myself. I'm Joseph Minicucci. I am the advisor for the American Institute of Chemical Engineers at Montana State University Student Chapter. And I have the pleasure today to welcome our two invited speakers, Joe Moran and MD Al-Azim. We're really lucky to have them here today. Both of them are currently working at Merck Pharmaceuticals, and it is a phenomenal opportunity during this global pandemic to have them speak and take time out of their day and on behalf of all of the students at AIChE at Montana State University and across the globe, Joe and MD, first and foremost, we want to extend our deepest thanks. This is a phenomenal thing, and your time is so valuable, and we really want to thank you for that. I know you didn't come here to listen to me, so I'm just going to introduce the two of you. In doing so, we are going to be able to listen to what you have to say about, basically producing and disseminating a vaccine in a presentation in 60 minutes. So our first speaker will be MD Al-Azim, who's a scientist in Merck's biologics division, or biologics process development group, excuse me, supporting international immunotherapy development and commercialization. And MD will pass the presentation over to Joseph Moran, who's a scientist in Merck Research Laboratories supporting vaccine development and clinical supply. And again, thank you, both of you, for being here today, and we very much look forward to hearing what you have to say. You'll be presenting for about 60 minutes, and then we'll have up to about 60 minutes for Q&A.
Fantastic. Thank you so much for that warm introduction, Dr. Minicucci. MD and I are so excited to be here with you and appreciate the invitation to partner with Montana State University to make this presentation and to have the opportunity to speak. And we're very grateful for that. And we're looking forward to having a very fantastic evening for those in the United States, and maybe a morning for those joining around the world. Can you see my screen, Joseph? All right, great. So with that, I'm going to pass it over to my colleague here, MD, and he's going to begin our presentation this evening.
Good morning, good afternoon, good evening to everyone joining us from wherever. Very excited to be here speaking with you guys. And to start off, my name is MD Al-Azim. As Joseph said, I support biologics process development at Merck. I have been with Merck for 3 years, where I supported mostly vaccine development during my time and vaccine manufacturing. So this is a very exciting topic for us to go over. So in this presentation, we're going to talk about the COVID research approach of different companies, their timelines, platform that they're using for their vaccine development, and some of the knowledge in the developmental activities, like process development, clinical trials, clinical campaigns. Then we're going to touch on commercialization. So going through process validation, process qualification. Then we're going to end with manufacturing, where we're going to touch upon different unit of operation that goes on in manufacturing. Next slide. So to start off, for the COVID vaccine pipeline diversity. So there are about 280 preclinical development of vaccine worldwide, which is a lot of vaccine that are coming. And as you can see in the x-axis, there's different phases that the vaccines are at, and the different color represent different platform. So blue would be the viral vector, which seem to be the most common. And then the reds are mRNA, green is DNA, purple is protein, subunit, and so forth, so on. And it looks like in phase 2 and 3 clinical trial, we have about 16 vaccines. So companies like Johnson & Johnson, AstraZeneca, Pfizer, Moderna, they're on the list doing some great work with their COVID vaccine development. So as I touched upon different vaccine platform, so what does it mean for it to be DNA-based vaccines or RNA vaccines or inactivated vaccines? So DNA-based vaccines would be synthesizing the virus DNA molecule, inserting it into a plasmids, and providing it into the human body, where the cells will take up the DNA plasmids and will make the viral protein that is capable of inducing immune response, and that's how it will provide immunity to human body. And Sanofi are taking that approach. For RNA vaccines, we have the antigen specific, so in this case, COVID viral mRNA sequence, where that will be introduced into the body, and that gets translated into the viral protein, which is also capable of generating immune response and provide immunity to human. And Pfizer and Moderna will be the one that are developing platform in this area. Then there is inactivated vaccines where the virus is being killed by heat or chemical means, and upon introduction to human body, it can't cause illness, but it is capable of generating immune response against the virus.
Hey, MD,
Mm-hmm.
before you continue, we're getting an echo. Can you turn your volume down, and Joe, can you mute while MD is speaking? Thank you.
Okay, is there any echo now?
No.
Okay, awesome. Thank you. Then the next one is viral vector vaccines. This is most common one that a lot of companies are taking this approach. It's almost similar to recombinant vaccines as well, where the viral protein sequence will be inserted into a host, or in this case, a harmless virus, which will be the means of delivery into the body. And upon introduction, it will generate immune response. And AstraZeneca, Merck, and Johnson & Johnson are taking that approach. Similar to this approach will be, as I mentioned, recombinant, where what they usually do is introduce the virus-like particle, which is don't have any virus genetic information, but it looks like virus, that usually gets introduced to hosts like yeast or E. coli. Then they produce the protein of interest, which gets purified, and upon introduction, that's also is able to generate the immune response against specific antigens. And as a virus-like particle, it doesn't have any virus content, so that's why it's not harmful. Then there is subunit vaccines, where a small fragment of the virus gets introduced into the body, and that small fragment is enough to generate immune response. And finally, unlike inactivated vaccine, there's also live attenuated vaccines, where the approach is to have the virus, but dilute the virus so it's weakened, and upon introduction to human body, it's able to generate immune response, and some cases it's better than the inactivated vaccines platform. Next slide. Now, here is an interesting slide about the vaccine investment and funding that different companies are receiving from the United States government, or Gavi funding, or European funding. And if you look at the y-axis, which shows the amount of dollar funding that they're receiving, Sanofi is, for their vaccine, they're receiving over $2 billion from United States government. J&J is almost 1.5 billion. Pfizer is receiving close to 2 billion. Moderna is also over $2 billion. And it's interesting to see AstraZeneca although they have 2 billion, but it's not entirely from United States government. Some of it also comes from Gavi and European funding. And there's other companies with their vaccine approach that are not getting as much as funding from the government agency as they are stated in the left or in the right. In this case. Next slide. So this is also another interesting slide where it shows that where these different companies are at with their vaccine approach. And in the x-axis here, you can see Q2 22, which means second quarter of 22, and Q4 22 is fourth quarter of 22. And it looks like the companies that are in their phase 3 clinical trial, which are Pfizer, Moderna, AstraZeneca, and J&J, they are planning to complete their phase 3 clinical trial by fourth quarter of 22. So Pfizer and Moderna are most likely going to complete it before J&J does, and then AstraZeneca will be shortly a little after. Next slide. So the next topic we wanted to discuss is the research and process development. Once you find your molecule of interest that is capable of inducing immune response, that's a great accomplishment at this point. But the next challenge comes up is that how can we make that molecule in larger quantity so we can supply them for clinical trials, and we can also supply them upon successful clinical trials. We can make them at a manufacturing setting, larger scale, so we can get to our patients and meet their needs. And most of these vaccine processes will involve an upstream and a downstream process. The upstream process would be where the cell culture that is capable of producing the protein of interest will be grown in a bioreactor, and then it will express the protein of interest. So the purpose will be to accumulate as much as biomass as possible in the upstream process. And the purpose of the downstream process will be to purify or isolate the protein of interest from all the other impurities and the host cell derived components, and completely be able to isolate. So that's both upstream and downstream faces its own unique challenge, which Joe will be going over later in his presentation. So in the scale-up process, there are challenges associated with identifying the proper unit of operations that are capable of doing both upstream and downstream related activities, evaluating the operating range. So what's going to be your temperature, what's going to be your pressure, what's going to be the agitation rate, things like that. That comes in very important, and defining those ranges so that if we have excursion from that ranges, we can control those parameters well enough every time consistently, so it would not cause deviation and would not have adverse impact to our final drug product. Then the next challenge goes with the sizing the equipment. So how big the tank should be, how many filter will we need to filter out our protein of interest from the impurities, how big our chromatography column needs to be. So those are some consideration goes into sizing equipment. And the sourcing equipment will involve with what vendors are we going to buy from filters, media, and column resin. So in these cases, the challenges would be, are the vendor capable of producing the materials that are in great quality? And not only that, are they consistently being able to meet our demands as well? So those are also some important consideration that goes into choosing that as well.
So now I'm just going to pass it over to me. I'm going to talk a little bit about clinical trials. And I'd like to begin by saying, what is a clinical trial? And it's going to be a study and investigation where we're going to be evaluating primarily in the beginning the safety, and then later in the later stages the efficacy of a new treatment. For our case tonight in this conversation, specifically a vaccine. Before we advance to clinical trials, there's going to be clinical stages which I will touch on moving forward. So depending upon the data that's generated through the clinical trials, if you fail phase 1, you're not going to progress to phase 2. If you fail phase 2, you won't progress to phase 3. So unfortunately, not every program that in development that begins clinical trials will complete them successfully and receive regulatory approval. But in the next couple of slides, we're going to talk about all the different items that go into clinical trials and how those outcomes are achieved. And specific to the pandemic that we're all experiencing, what might that look like moving forward when we think about the slide that MD presented, listing different companies and their different stages of clinical trials and when they're expecting to complete them. So here we have a nice image talking about different phases of clinical trials. All the way on the left, we're referencing discovery time, where we're going to be conducting.
Hey, Joe, I'm sorry to interrupt. You're fading in and out on your microphone. I don't know, maybe you'll have to speak up a little bit more on that.
Thank you. Is this a little bit better now?
A lot better, thank you.
Fantastic. So all the way on the far left, we have discovery science, where we're going to be starting with lab studies or lab experiments. And then we're going to progress into preclinical trials, which are going to be executed in animal models. And then after that, we're going to move to our first human patient. And if we see that the vaccine is well tolerated, we're going to progress to phase 1 clinical trials, which from a clinical development perspective in research is where your efforts are really going to intensify. And at that point, you're going to be enrolling a small number of patients, maybe 10, 20, 30 patients. And you're primarily going to be interested on demonstrating successful safety of your vaccine. There may be some changes or variation in the concentration of the dosage of the active ingredient, but primarily you're just looking to generate that nobody's going to be suffering any significant side effects. And that might take maybe 6 months, after which you're going to expand your population of patients and you're going to further evaluate the safety. Safety will always be a consideration of your product, but now you're also going to start moving into assessing efficacy and does it generate successful immunogenicity data. And then after phase 2, you're going to progress into the final stage, phase 3, which could be up to 2 years. I think there are a number of companies are around that 2-year mark with their phase 3 clinical trials. And in the subsequent slides to follow, I'm going to be connecting, what is the significance of that in terms of global access to potential vaccination. And here you're going to be drastically, exponentially expanding your patient population to upwards of 20 or 30,000 patients enrolled across a variety of different demographics and ages to encompass and truly represent the general population of the global public. And as you're executing phase 1, phase 2, phase 3, you're also going to have patients that are injected with a placebo. It might be a saline solution, which is going to have no effect. But for the purpose of a successful clinical trial and making sure that you're not favoring advantageous results, that's also going to be a component as well. So now I'm going to talk a little bit about how clinical trials are supplied, how that supply partners with process development and developing unit operations and sourcing materials like MD spoke upon a few minutes ago. And I really want to emphasize the importance and the criticality of a working virus seed campaign. And I'm going to talk about what that is. And I also want to make sure that I emphasize the differences between phase 1, phase 2, and phase 3 material, and what you are and what you are not allowed to do or change as you're pursuing those clinical supply activities, And if you do start making too many changes, then you might need to have further studies or further experiments to demonstrate comparability and show that what you supplied in phase 1 is the same as what you supplied in phase 3, even though you may have changed a number of variables. So starting with a working virus seed, what is a working virus seed campaign? It's critical to the success of a program. Let's say, MD and Joe decide to make their own COVID vaccine, and we select E. coli as our cell line. We're going to grow a lot of the E. coli bacteria. We're going to reproduce them, and then we're going to, insert a plasmid, and that's going to express our DNA-based vaccine. So in a working virus seed campaign, we're going to be interested in going through the cell culture and fermentation process for E. coli, increasing the number of E. coli cells, which will be talked about in more detail later on. What does fermentation look like? And then after we accumulate that biomass that MD was speaking about, that's when we're going to be infecting our cells. And depending upon what our virus is, and depending upon what our cell line is, the behavior that we see at that point could be different. We might see the protein of interest secrete out through the cell wall into the supernatant of the bioreactor. We might see it captured in between two cell membranes, which would be called a periplasmic space. We might see it stay inside the cell, and then we're going to need to either lyse the cell with a homogenizer or maybe some kind of specific buffer that's going to cause the cell wall to break or rupture. But there are going to be different strategies that are used there depending upon what your cell type is and how that cell type responds to your virus. But for a working virus seed campaign, it's at that point you're going to stop, you're going to pause. You're not going to pursue purification. You're going to capture your virus material at that point, and you're going to have it dispensed out into different sample containers for long-term storage. At long-term storage, you're talking about significant low temperatures. Could be -70 Celsius, could be -160 Celsius, depending upon what your cell line is and what the virus stability is. And the reason that you're going to have an interest in those very low temperatures is to make sure and you're minimizing any opportunity for the stability to be affected of what you've captured and dispensed, and to make sure that you're minimizing molecular movement. So you're pretty much just preserving that over time. And then if you were to be successful and go through the commercialization and arrive at manufacturing, those working virus seed containers, each time you go to manufacture your vaccine, or MD and I would be doing our E. coli based vaccine, we would be using one of those containers to infect the E. coli cells that we're growing at the production scale. So a working virus seed campaign is critical to the success of any program, and ensuring that you're going through the proper quality control and release testing to make sure that you don't have any contamination is also very important. And that's going to be something that continues through the life cycle of the product. If MD and I were to be successful with our E. coli COVID vaccine, maybe 10 years from now, we're starting to consume all of the working virus seed. At that point, we would need another working virus seed campaign, which we would just repeat with one of the previously dispensed working virus seed containers, and we're just going to generate more material all over again from the same virus line. So that's what a working virus seed looks like, and that's why it's so important. And it's going to be pretty much the life cycle of your product. And now we're going to talk about how that's going to contribute to different clinical trial campaigns, starting with phase 1. So if you remember from the previous slide, a phase 1 study is going to have a handful of patients, maybe 20, 30, 40, 50 patients. So you're not going to need hundreds of kilograms of material. So your unit operations might be smaller. You might not scale up a bioreactor to 5,000 liters. Maybe you're working in a 10-liter bioreactor. Maybe you're working in a 50-liter bioreactor. And then depending upon the size of bioreactor that you're working with, that's going to have an influence on the surface area that you need for filtration, and the size of your filters, and the column volume, and the amount of resin that you need in a chromatography recovery step, as MD touched upon earlier. So your phase 1 campaign is going to be much smaller compared to your phase 3 bulk campaign to supply phase 3 clinical trials, where you might have 30,000 patients and you might need 30,000 doses. So maybe now you're looking at several hundred kilograms of material coming out of your bioreactor that need to be purified. And between when you start or go through your phase 1 bulk campaign compared to your phase 3 bulk campaign, generally speaking, in quote unquote normal vaccine development, that could be over a period of 2 or 3 or 4 or 5 years. For COVID vaccine development, which has accelerated timelines and massive amounts of funding invested, you could be overlapping significantly by saying, well, we're going to have a phase 1 campaign in, maybe we're going to have a phase 1 bulk campaign today, and then in December or January, we're going to be moving forward with a phase 3 bulk campaign. So as those changes are happening and the scale of your process is changing, your process development group is also going to be partnering to do the best they can to advance the different variables for different unit operations. MD touched upon maybe pH or temperature or agitation rate, and to optimize your process to maximize what your yield is. So, you could change your agitation speed, you could change your temperature, but if you start saying, well, now we're going to use a different kind of media that we didn't use in phase 1, but we're going to use that media in phase 3 because we get much better growth in the bioreactor, that might be something that starts moving into that consideration for, do you need a bridging study to demonstrate comparability because you're using a different media now? So that's an example of something that might be changed through process development over a course of time that would require a bridging study. Here we go with a lot of information, and it's all about regulatory approval. So regulatory approval would come at the completion of phase 3 clinical trials. You get your data set back, that's submitted to the FDA for their evaluation, and assuming that you successfully demonstrate safety and efficacy in your broad patient population of maybe 30,000 patients, you can expect after between somewhere around 6 or 10 months of review, the FDA could come back and say, we approve you and you now have regulatory approval. So specific to the pandemic that we're all experiencing, if you remember MD's slide where he talked about maybe the 4th quarter of 2022, when maybe Moderna or Pfizer or J&J would be completing their phase 3 clinical trials, regulatory approval would be, the review process would be started at that point after that data is all collected. So how would we have a vaccine available for use if we're not going to complete phase 3 clinical trials until 2022? And that's where we start leaning towards the left side of the slide here, where we're talking about emergency use authorization or accelerated approval. So in an emergency use authorization experience, maybe the FDA is saying that based upon some intermediate points of the phase 3 clinical trial, maybe it's at 3 months, maybe it's at 6 months, but it's at some defined point of progress based upon the information that we have. So far, it looks promising, and the immediate need that we see for patients, we're going to move forward and authorize emergency use authorization, which means that it's going to be available to the public. And that's different from accelerated approval, where you're likely predicting the final outcome of a clinical trial. If you have emergency use authorization, there's a chance that you won't make it to accelerated approval. If you have accelerated approval, there's a chance that you won't make it to regulatory approval. But those are intermittent avenues that could become final avenues to regulatory approval and promote the opportunity for market access for patients in specific circumstances. I think the global pandemic we're experiencing for COVID is one of the best examples we might ever see of a need for emergency use authorization or accelerated approval. So I wanted to make sure that I, MD and I collectively in this presentation spoke specifically to what those differences are and how those different opportunities for approval could have an influence on what we all might see together in terms of a vaccine being available. So now I'm going to pass back over to MD, and he's going to talk about commercialization. What is commercialization, and what does that entail?
So commercialization, it's a very fun place to be in. Right now, at this point, you are getting some good data in the clinical studies where your molecule of interest is working, and you would like to think about now making it in a larger scale in a commercial facilities. So the things that you need to consider during this time is the FDA process validation stages. There will be 3 stages in the commercialization. At stage 1, which will be finalizing the process design, in this case, the process is almost locked depending on the development activities and the scale-up activities that took place in the lab. We have a very good understanding of our process. We have good understanding of all of our critical process parameters, critical quality attributes, and we are finalizing the process design to be able to commercialize in larger scale. In process qualification, which is stage 2, there what we're trying to accomplish is to see whether or not we are capable of making the same batch or making the same product with meeting all the critical quality attributes and the important parameters. In this case, we will try to make 3 batches and showing that each and every time we were able to manufacture the batch with the same quality and met all the required attributes for us to say that, yes, our process is robust and we are capable of making the process without having any significant deviation or excursion in the process. And stage 3, that will be once the process is validated and we are making the process, it's an ongoing thing that at some frequency, whether it's 6 months or a year, we would be continue to evaluate different trends in our manufacturing process, looking at our critical quality attributes and seeing how they're behaving with respect to time. And we will do statistical process analysis in this case to identify any outliers and what might have causing this to bring our process back to in a state of control. Next slide. So as I touched upon critical process parameters, these are the parameters that if we have an excursion in those parameters, that can significantly impact our critical quality attributes. So we want to have a process control in our manufacturing setting to ensure that we are capable of meeting our critical process parameters every time. Now, if in regular manufacturing, if we have a violation of critical process parameter, in this case, there would be a lot of extending testing would go on and a lot of sample would be taken to ensure that we met all of our critical quality attributes prior to making a decision on whether or not we're going to continue the batch or we're going to discard the batch. Next slide. So critical quality attributes are physical, chemical, and biological properties of our final product that will define the safety, efficacy, stability, and quality of our product. So we want to ensure that this critical quality attributes, all of them has to met at all times before we can release this particular batch to the filling and formulation from a drug substance, and then finally to be able to deliver it to the patient. At each stage, we would go through rigorous testing to ensure that all of our critical quality attributes have been met. Next slide. So here is an interesting slide that shows the control strategy, design space, and acceptable ranges, tying together with critical process parameter, and also some financial concern there to optimize the process the most effective way. Where we ideally want to be in the manufacturing region is at the green square would be the optimal one, and that would be a dream place to be in, because in that place is where our process is optimal, our process is robust, and our process is in a state of control where we are able to make the same quality of product every time at low cost and being able to deliver that to our patient. Next slide. This is another slide that I found to be very interesting. So the risk metrics is what usually being considered to identify what are some parameters that are going to be considered as critical in the process. So each and every single parameter of a unit of operations will be evaluated based on 3 questions. What is the impact on purity if we do not meet that range for that parameter? How likely will this to occur during the manufacturing? And what is our ability to detect this variation during regular manufacturing? So based on that, we would assign some points to that. As you can see, for the purity one would be minimal, 5 will be moderate, and 9 will be significant. Similar for probability question, 1 is unlikely, 5 is moderately likely, and 9 is high likely. And the ability to detect, 1 is certain, 5 is moderate, and 9 is unlikely. So based on this question, there will be number given, and if we multiply all 3 numbers, we will be able to obtain the risk probability number. And the higher the risk probability number is, the most likely that parameter will impact the quality of our product. So that's how we would say that this parameter is critical parameter for the processing. So in this example, it shows the unit of operation is crystallization, and it's evaluating temperature, water content, it also addition time, anti-solvent percentage, agitation, and giving those risk number to identify what would be the parameters to look out for.
So with that, we're going to move back over to me, and I'm going to conclude over the next 15 to 20 minutes our presentation as I speak about manufacturing and what that process entails from start to finish, all the way from upstream cell culture to labeling, packaging, and making that available to the patient. So here we have a nice slide that encompasses the product life cycle. If you start on the top left with cell culture and move across the top line to the right, we're going to progress through inoculation, and cell culture and inoculation together are going to encompass upstream or encompass fermentation, harvest, depending upon what cell line you're working with and what the behavior is. Like I touched on earlier, are you in the periplasmic space? Are you in the supernatant of the bioreactor with your protein of interest? What does that look like? Harvest could land in either fermentation or purification, so continuing across the top to the right, or we have bulk and purification listed. That's really going to be where we're moving to recover our protein of interest, as MD described earlier. And as we progress down to the bottom, underneath purification, we're going to have formulation, where we might be using different stabilizing buffers that might help us to have stability at different temperatures. We might be including an adjuvant. I'll touch upon what an adjuvant is as I go into this slide in more detail. And finally, from formulation, we'll move to filling, inspection after completion of filling, and then finally labeling and packaging your product, at which point you're going to be distributing your product throughout the supply chain. So starting with cell culture and inoculation, for MD and I, if we're using E. coli to generate our vaccine, E. coli is going to be our cell line. So we're going to have a master cell bank of E. coli, and we're going to take a few milliliters of that that's frozen, thaw it out, and we're going to start our cell culture process, which could involve different types of media. Now, depending upon what your vaccine construct is it a DNA-based vaccine? Is it an RNA-based vaccine? Is it a viral vector? That slide that MD touched upon at the beginning of our time together, that's going to have an influence upon what cell line is going to be the most advantageous. Maybe you're going to be interested in using E. coli or bacteria cells, or maybe another type of bacterial cell. Maybe you'll be interested in using a yeast cell. Maybe you'll have an interest in a mammalian cell. Maybe you'll see better growth or better protein generation in one cell line compared to another. You might be evaluating different cell lines in the discovery science or discovery research component or area before you move forward with your clinical campaigns. So depending upon what cell line you choose, you could have a bioreactor where your cells are suspended and they're just in media and you're agitating them. Or you could have cells that are adherent and they need to be attached to some kind of substrate in order to grow. If I'm understanding this correctly, that image above harvest is a number of roller bottles. So I'm thinking that that's most likely an adherent cell line, and where you see those red lines is the media where the cells are going to be adhered to the bottom of those flasks or bottles. But there are different strategies that you're going to be using depending upon whether or not you're working with an adherent cell line or a suspended cell line. Suspension is preferred. Suspension is easier to control. And a strategy that you might be using if you're working with an adherent cell line is to introduce microcarriers. So a microcarrier is a small bead. We're probably talking about microns here in diameter. I think a human hair is 100 microns in diameter, to put it into perspective. So we're smaller than the diameter of a human hair with microcarriers. And those are going to be mixed into your media. And then your adherent cells are going to be charged to your bioreactor, and they're going to attach to your microcarriers. And that's going to allow them to be suspended in your bioreactor, but also have the adherence that's required for them to continue to proliferate and grow. As we're going through the fermentation process, we're going to have an interest in temperature, we're going to have an interest in pH. Depending upon what temperature or pH we're operating at, we may express different impurities. So, maybe if MD's in the fermentation group and I'm in the purification group, and they decide they're gonna grow their cells at 30 degrees Celsius, maybe I'm saying I have all of these impurities now that I need to remove, and I need to use all of these different purification strategies, which we're going to be moving towards in a few minutes. But maybe if they change their temperature and they go and grow their cells at 20 degrees Celsius, or maybe they change their pH, all of a sudden those impurities might disappear. And that eliminates the need for additional purification or unit operations moving forward. So depending upon what decisions you make in fermentation, what your cell line is it suspended, is it adherent, what's your pH, what's your temperature, what's your dissolved oxygen concentration, how are you balancing the different operating parameters of your bioreactor. Maybe it's agitation, maybe you're charging carbon dioxide to influence your pH, maybe you're using sodium bicarbonate to make it more basic. If you use sodium bicarbonate, if you use too much, now are you having those sodium ions influence your cell behavior? So there are a lot of variables that need to be managed to successfully grow those, for us in our example, E. coli cells, to accumulate that biomass so that we can then inoculate and introduce the virus, which in the manufacturing world is going to come from that working virus seed campaign that I spoke to earlier, to ensure consistency and there's no variation there. So when we complete fermentation and we're going to go to harvest, we might be using an osmotic shock buffer, we might be using a homogenizer to recover our protein of interest, whether it's in the cell, whether it's in the supernatant, at which point we're going to be then beginning our bulk purification process. And there are a number of different strategies that are going to be required and influential to be successful there. Beyond just your normal unit operations, there's going to be significant interest in buffers, and depending upon what your process looks like, you could be working with up to 5, 10, 15 different buffers that might have different salt concentrations, that might have different pHs, and have all been targeted to a specific range or a specific value to make sure that you're being successful in the unit operation that you're working with. If you're going through some kind of filtration step, you might have a buffer filtering into your product tank, which is then carrying it along through your filtration. So in theory, it might be diluting it, but that might be presenting an environment that's more desirable for either additional recovery later on, or for maybe to become either dissolved in solution or precipitate out of solution to either pass through your filter and be in the permeate, or maybe you want it to not pass through your filter and stay on the retentate side. So depending upon what your pH might be, that might be an opportunity for you to influence whether or not you're precipitating out your product. In addition to filtration, we might be working with some kind of chromatography step where we could have a chromatography column with a resin inside. It might have an ionic charge. It might have, it might be a size exclusion column. So there are a variety of different chromatography column separation techniques. For MD who might work with monoclonal antibodies, who might be using a protein A resin, which is very expensive. So at the manufacturing production scale, now you have a significant interest in trying to reuse that resin, recover that resin to maximize the yield that you're generating with that column. And those could be additional studies that might not be relevant or might not have an opportunity to be executed in an accelerated vaccine development timeline for COVID and the pandemic, but after which you receive, maybe regulatory approval if you were to complete clinical trials. Maybe now you have a chance to go back and perform those reuse studies for your resin and update your filing and then validate that process so you can reuse your resin or maybe reuse your filters to maximize, from a business perspective, the value that your product is offering. In addition to chromatography and filtration and buffers, we're going to have a dispense area of purification. And if you're looking at a supply chain where fermentation occurs in one country and purification occurs in another country, and then maybe formulation occurs in a third country, how you dispense your purified product is going to be important. Maybe if you're dispensing your product into 30-liter containers and your formulation suite that's in Europe, the purified products coming from America and moving over to Europe is going to be 320 liters. You're going to need 11 30-liter bottles to fill that formulation tank, and you're going to have potentially 10 liters of product left over, which might be wasted. So how can you effectively construct, evaluate, optimize your different processes, fermentation processes, purification process, formulation process to maximize your yield, minimize product lost as you're transferring from one facility to another? So moving forward into formulation, we receive our bulk drug substance. Now we're interested in turning it into drug product, and we want to make sure it's stable. We might have an interest in saying we're going to use a buffer that's not sensitive to light or photosensitive, so that way we don't have to protect it from light. Light is an opportunity for degradation to occur in a protein, so how might we minimize that variable? Additionally, besides a stabilizing buffer to prevent any degradation of our protein, we might have an interest in an adjuvant. And what is an adjuvant? So an adjuvant is something that might be included or mixed in. It's another molecule, and collectively it might bind to the protein of interest. So that way, when a patient receives an injection, maybe your initial immune response is specific to the adjuvant, and then upon your immune cells arriving at the adjuvant or identifying the adjuvant, that's when they discover the protein of interest specific in our discussion this evening to a vaccine against COVID. And what is the advantage of using an adjuvant? An adjuvant might allow you to have a lower concentration of your protein of interest in your final formulation for your vaccine, which might then allow you to generate more doses for the same amount of drug substance that you produce. And now you might be sitting there saying, well, what about the adjuvant? Now you have to make the adjuvant. And depending upon what your adjuvant is, it might also need a fermentation process and a purification process. Or maybe it's some kind of precipitation reaction or chemical synthesis, which is significantly faster than your protein of interest process. which allows you to maximize your output of your product. So an adjuvant is an opportunity to potentially increase the number of doses that you're making. Some vaccines might use it, some vaccines might not, but it's an option that's available. At which point, when we conclude formulation and we have a homogeneous mixture of maybe our protein of interest, our stabilizing buffer, and our adjuvant, now we're going to move into filling. And filling, it can be overlooked because you're not going to have any fancy separation techniques or any fancy variables that you're controlling in balance with each other in fermentation, but it could be the most critical part of your life cycle because this is your most expensive product. Your product has more invested into it at this point than it does in purification or it does in fermentation. So if you were to have a discard here, this is going to be, a bigger loss to either the business or the patient population in terms of constrained supply. So when you move into filling, you're going to have the option to go with a pre-filled syringe or a vial where you're going to be drawing the dose out of the vial into the syringe. The advantage from an optimization perspective is to go with a pre-filled syringe. The reason why is when you use a vial and you draw the dose out into a syringe, you're going to have a remnant left in the vial, and maybe your target dose is 0.5 milliliters, so your vial might contain 0.7, and upon drawing out your target dose into the syringe of 0.5, you might have 0.2 left in the vial, and that gets discarded. From a percentage perspective, you might be able to manufacture 20 or 30% more doses with the same volume of drug product that's been formulated if you use a pre-filled syringe instead of using a vial. So now as we move from filling, we're going to talk about inspection. What does inspection mean? So now that we filled our product, we need to inspect our either syringes that are pre-filled or vials for defects to make sure that they don't contain particles, to make sure that they're not cracked, to make sure that the stopper is correctly positioned, the plunger is correctly positioned, and that the patient is not going to receive a compromised product. So your inspection is going to most likely be automated, especially when you're talking about a scale required to vaccinate a global population, where we're talking about hundreds of millions or billions of doses. And in order to do that, you're going to want to have a very conservative automated inspection system. So you're going to have rejects. Maybe you're going to be rejecting 5% of your product as a reject, which will not go to market. And, of that 5%, maybe cumulatively 10%, which might be 0.5% of your total product, is actually defective. But you want to be conservative. You want to minimize the opportunity for a compromised syringe, a cracked syringe, a vial with an incorrectly positioned stopper to make it to the patient. At which point, when you complete inspection, you're going to move forward into labeling and then packaging, at which point you're going to move into distribution. There's a strong opportunity or a strong likelihood your final product is going to need some form of temperature control, depending upon what your virus is. Like MD talked about at the beginning, is it a viral vector? Is it a DNA-based vaccine? That selection could have an influence on the temperature that you're going to be storing your packaged product at. Maybe it's slightly below freezing, maybe it's slightly below room temperature, but you're going to have different supply chain needs for storage and distribution depending upon what temperature you're going to be looking or operating at. If you need to store it at -60 or -70 degrees Celsius, now you're going to be shipping it and distributing it on dry ice to make sure that you're being successful with that. So those are some different details relevant to the product life cycle: fermentation, cell culture and inoculation, purification, harvest, chromatography, filtration involved there, formulation, where we're talking about maybe using an adjuvant, maybe introducing a stabilizing buffer, filling with vials or syringes, inspection, a reject rate, and then finally your final packaging and what your long-term storage might look like. And before we conclude, I also want to touch upon the influence of single-use technology and cleaning validation and what do those words mean, and how do they contribute to exit cycle time, and what is the significance of exit cycle time. So exit cycle time is going to speak towards the number or the amount of product that you're generating from your facility. So let's say we're looking at the purification step here of the product life cycle. If it takes me 24 hours to manufacture a batch of purify a batch from start to finish, and if I start a batch and then I let that batch finish before I start another batch, I'll be pushing out a batch every 24 hours. If halfway through at the 12-hour mark, I start a new batch, now my exit cycle time becomes 12 hours, because every 12 hours I'm starting a batch, every 12 hours I'm going to be generating another batch. So that's an opportunity for you to overlap different batches and move more product through the facility to make it accessible to patients. Single-use technology might allow you to avoid equipment turnaround, which might be using steam or other cleaning agents to clean your pipe network, to clean your unit operations, to make sure that it's ready to receive your next batch, and you're not going to have any cross-contamination. If you move towards the single-use components, which after you use them, you throw them away, now you might be able to reduce your exit cycle time beyond that 12-hour mark and generate even more product. So that concludes all the different variables and all the different components and pieces of vaccine development, commercialization, manufacturing that we had prepared. We're very grateful, and we want to give a special thank you to Dr. Menachcucci, Austin, and Madison at Montana State University for the invitation to present tonight. We hope the material that we covered was beneficial for our audience. We appreciate you joining us. And with that, we'd like to include a little bit of humor in our presentation this evening, and kick it off with any questions for those of you who might still be joining us and still be awake. So thank you very much.
Thank you very much, Joe and MD. I'll kick off. Anybody listening, you can submit questions through Q&A down at the bottom of your screen. And we have already 5 questions coming in, but I'm going to ask the second one first, because I think it gives you some time to warm up here a little bit for both of you. Can you tell us a little bit about your backgrounds? What are your undergraduate degrees? Do you have graduate degrees? And what do your paths look like along the way to getting to where you are?
Sure, so I'll go first. I have obtained my undergraduate degree in chemical engineering from Manhattan College in New York City. And during my time, I always had a high level of interest to have a career in pharmaceutical. I'm originally from Bangladesh, and being from a country like Bangladesh, where there's not a lot of opportunity for medication and treatment and things of that nature, that actually what drove me to pharmaceutical industry. As an undergraduate, I had internship and co-op with Merck. I really enjoyed my time working at Merck, so that when I graduated, I took a full-time opportunity at Merck, where I found a lot of value in my time at Merck for the last 3 years. I was involved in 2 different vaccine manufacturing. In my time here, I have done vaccine manufacturing for HPV and hepatitis B. Both are very big life-saving product. So it was definitely a good experience to be part of their franchise. And currently I'm working with Keytruda process development. So that's also another rewarding experience that I'm enjoying. And currently I'm also going to grad school. In addition, my time with Merck, I'm currently enrolled in Johns Hopkins University to get my master's in biotechnology.
So coming over to me now, similar to MD, I also graduated with my degree in chemical engineering in 2016, and I've been with Merck for the past 3 years now. My current position in Merck Research Labs is my 3rd role. My previous 2 roles, my first one was at the Elkton, Virginia plant site supporting the purification of Gardasil 9. The 9 is specific and interesting because that speaks to the number of serotypes involved in that vaccine. So we would have to make 9 different batches, one for each serotype, before they could formulate their final product to make it for the patient. And upon developing and learning about different unit operations and identifying what my career interests might be, I decided to relocate to West Point, Pennsylvania with Merck and join another area of their supply chain where I was supporting formulation and filling, not only for Gardasil 9, but also for Combavac, which is actually the vaccine that MD was then purifying and sending over to my group. And then also Vacta, which is hepatitis A, and Pneumovax, Pneumovax 23, which prevents against pneumonia. So that was a tremendous opportunity for me to have different exposure to other vaccines in Merck's network, learn about other vaccine processes, how they might be similar to Gardasil 9, how they might be different. And collectively, those experiences were critical for me to leverage as I transitioned into my new role as a scientist in research labs. I now have the opportunity to support multiple programs going through development, and one of which contributes to the involvement that Merck has in developing a vaccine to address COVID and hopefully become commercial and accessible to patients. So I'd say in the 3 years that I've been with Merck, it's been a tremendous learning experience coming out of chemical engineering in 2016. If you asked me about pretty much, I'd say 95% of the material that we addressed tonight in this presentation, I never would have known about it. So I hope that, to our audience, you guys find this valuable. And if you have a specific interest in career in biopharmaceuticals moving forward, maybe this is an opportunity for you to leverage that knowledge moving forward to achieve your own interests. Similar to MD, or identical to MD, I'm also enrolled currently as a graduate student at Johns Hopkins, also pursuing my master's degree in biotechnology. I decided, just like MD did, that would be a tremendous opportunity for me to complement my command of chemical engineering principles that I developed as an undergraduate with enhanced biological understanding to suit my future career interests, pursuing additional responsibility to have a fulfilling impact, specifically in large molecule biopharmaceuticals.
All right, excellent. So we're going to move to the next question from Maria, and they ask, when companies do clinical trials, they have different phases. Who determines the phases, and do they vary on the country, or are they worldwide?
So I'll go first, and Joe, you can chime in. We're not an expert by any means in clinical trials, but to best of our understanding, I believe that phase 1, phase 2, and phase 3 clinical trials are consistent throughout all different regulatory agencies around the world. I don't know, Joe, if you think that it might be different. I think it's very consistent, and it's very like similar, that you have to go through all these 3 different clinical trials for you to prove that the vaccine is safe, it's stable, it can generate immune response, and can do what it's targeted to do.
So, MD, thank you. I think that's a fantastic, beginning to the explanation. To go into a little more detail, I'll say that generally speaking, phase 1 clinical trial or phase 2 or phase 3 clinical trial, you'd only need to execute one time. You wouldn't need to execute a phase 3 clinical trial for each company that you're interested in registering your product. So for Moderna or Pfizer or J&J, some of the major companies that we explained earlier were in phase 3, they're going to need to execute one phase 3 study, and then based upon that data, they're going to need to submit to different regulatory agencies that may be country specific, the FDA being one for the United States, the European Medical Agency being the one that might be governing Europe, the China FDA, which might be governing China. So for different markets and different patient populations across the world, you might need individual regulatory filing, which is going to be reviewed by that specific regulatory agency, but you're not going to need to do additional clinical trials specific to each regulatory agency.
Okay, Austin looks like he's muted, so I'm going to jump in here for a second. I don't know what's going on there. So we have a question. What are some actions taken in the case that your virus fails at one point in time at a critical parameter or a quality? Is the entire batch scrapped, or is there a way to salvage it and reduce losses?
That's a very good question. That's something that manufacturing world sees a lot, I would say, and it's a huge challenge, right? Like every time you have an excursion with your critical process parameter. So I guess the consideration will go towards, okay, like let's say your temperature is out of range of chromatography process, right? And your chromatography is intended to purify the host cell protein, high molecular weight species, and some viral clearance there, and let's say some DNA clearance. So it's doing a lot of clearance for us. The temperature is outside of the range. What will happen next? So what we would do at this stage, we would take extra sample to have testing done for the host cell protein, for DNA, for high molecular weight species, and see whether or not we are meeting those acceptable criteria. If we do meet that acceptable criteria, which we define as our critical quality attributes, in that case, and we have strong enough technical justification as to why it won't adversely impact the quality, purity, safety, and efficacy of our product, then it will be good to proceed. But if we don't have enough technical justification for those things, and we can't show that we are able to meet our critical quality attributes, that case, the batch will be discarded.
So that's a fantastic response from MD. To add to that, which he spoke about manufacturing in the clinical space, if we're going through maybe a phase 3 campaign that's going to be supplying a phase 3 clinical trial, and there's a question about product impact because we're outside of our temperature range, or maybe our pH in the bioreactor was compromised for a window of time that might be 10 minutes, it might be 10 hours, that's most likely going to move towards an immediate discard to eliminate any potential risk. And then that's going to be re-executed to be successful.
Yeah, I think we're losing Austin here. Is that, can you hear anything, Joe? Okay, Madison, can you take the next one?
Yeah, I got it. So somebody asked, what do you see as your role in maintaining ethical standards in the pharmaceutical industry? And how, if at all, does that influence your day in, day out work?
I would say that working for a company like Merck, who takes patient safety very seriously, and I think what I really like about Merck is their mission statement. One of the founders, George W. Merck, who had said that medicine is not for profit, it's for people. And if you do the business the right way, profit will follow. And I feel like, the company lives by this statement. And on a day-to-day basis, whatever decision that we make with our product and with our effort, we try to give priority to our patients and try to always keep that in mind as we are doing our day-to-day activity. And it's very rewarding to be in this business, right? Every morning waking up, going to work, to us, it's like we're saving another lives and buying extra time for people through the novel vaccines or biologics that we make every day.
I'd like to add to MD's response, and I'd like to thank our audience member who brought that question forward. That's a fantastic question, and it's a very broad question. So despite MD having a tremendous response that is perfect, there's also other areas to touch upon. And I'd like to say that at, I think, any biopharmaceutical company, regardless of whether it's Merck or maybe one of our competitors, from our perspective, maybe Pfizer or J&J, we'd like to see them do well, but we're also competing with them. And any major player is going to have strict regulations, and he's going to have electronic documentation and an emphasis on data integrity to defend and support the training of their employees, to make sure that qualified people are involved in the behaviors that they're executing, and also in terms of, you know, stability or quality control data that's going to be required in order to release a batch to patients. So that's something that is scrutinized at the highest level of, I'd say, any organization. Specific to Merck, we're going to have internal audits where we pretend we're a regulatory agency and we're performing a regulatory inspection to challenge ourselves to have a high standard. And then we're going to be hosting regulatory agencies, not only the FDA, but we might have people travel from China, we might have people travel from Russia, we might have people travel from Saudi Arabia, different countries that we have filings in for different products, to come and inspect those processes, come and inspect our documentation, and make sure that we're meeting the standards that they require. That might happen on an annual basis, it might happen on a biannual basis, depending upon the regulatory agency.
All right, transitioning into a different area, into production. Specifically for DNA-based or viral vector vaccine, which step in the production lifestyle is the most difficult in the development before you reach clinical trials?
That's a fantastic question. I would say that, you know, dealing with recombinant DNA or dealing with viral vector, they both have their own challenges and advantages. So the DNA based would be, it's a lot common, the recombinant DNA approach, that happens a lot. It's a very well-known technology. So is to some extent the viral vector. But the challenges with the viral vector would be that you are using virus as a mean of entry for your molecule of interest to be delivered into the body. So there will be some concern of how is that virus that you are utilizing to deliver the vaccine is going to have an impact in the human body, and whether or not that virus is capable of causing some adverse effects that could cause some serious illness. So that's approach needs to go through a lot of studies, and they need to demonstrate the safety of their processing, where the DNA-based approach, the main challenges would be to be able to produce an antigen that would be effectively produce the immune response to the best way possible. And in some of the cases, what they do is they also add some type of adjuvant to it, as Joe touched upon, to be able to induce strong immune response to that. So I think they both are very different in their own way. I would say that based on my experiences, I have worked with recombinant DNA. I don't have much experience with viral vector vaccine. I would say that recombinant DNA would be more easier to handle. I could be biased because my experience was with that.
Joe, did you have anything to add on that one, or shall we move to the next question?
No, I'd like to add to that. Fantastic response, fantastic question. I'm going to say that it's not going to be product specific. So if you're manufacturing a vaccine that might be recombinant DNA, maybe you're using a cell line that's very robust, very healthy, can withstand fluctuations in temperature, pH, agitation. Maybe you're not going to have a lot of concerns there. Maybe it's very well defined and it's previously been used in other products and it's not very experimental. Maybe you're using something that's an adherent cell line that might be difficult to grow. Maybe you're using microcarriers. Maybe you don't have the most experience with microcarriers, and you know that could be a challenge to be successful with. I think a very important component or variable to consider in this question is, how do the decisions you make influence yield? How do they maximize yield? I think it's easy to say we're going to have a process that goes start to finish. I think it's very difficult, very challenging, might be almost impossible to say we're going to have a perfect process that gives us the maximal or optimal yield. So I think, you know, as you consider different unit operations or different areas of your process, either upstream fermentation, purification, downstream formulation or filling, you're most likely always going to have an area to improve. Maybe it's something small like a filter reuse. Maybe it's something more significant like capturing more product on a chromatography column and increasing your yield. So you know, I think that it's a tremendous need for an engineering perspective and an engineering mindset, and you know, hopefully through rigorous academic performance and then increased industrial experience, different people are going to be qualified to collaborate together. I'd say that another thing that I've learned through my time at Merck is I can't do everything. I can't do purification and fermentation and formulation and filling. So the need to be a team member and say, I might be able to accomplish a lot as an undergraduate or a lot in my school, but when you start looking at how can you answer that question of, which part of the process might be most difficult, that's going to be a collaborative approach. And collectively, if you have an army of engineers working together, you still might not be able to answer that perfectly.
I think we have a question here that might follow this up. Matthew asks, and Matthew's done his research, it seems. As he understands it, Merck is developing 2 vaccine candidates, one that uses an RVSV platform and one that uses measles virus as its vector. Why were these platforms selected as opposed to other platforms, and why not attempt a completely different type of vaccine, such as an attenuated virus, instead of one of the candidates currently in development?
That's a fantastic question, Matthew. Great job in doing all of your research there. I would say that, of course, like there were consideration of all different platforms, and I'm not involved in making this decision, so I can share what I think had happened, or how did we decide to go with these 2 vaccine approaches. We did recently had made vaccine using the similar platform. So we do have experiences, we do have the capacity and the knowledge to be utilizing those platforms instead of using the other platform. And I think that was one of a variable of many that went into the consideration. Of course, there are a lot more consideration had gone on to be defining that these are the platforms that we're going to be using.
To build upon MD's response there, I think that when you start looking at that emergency use authorization, accelerated approval versus regulatory approval experience over a phase 3 clinical trial, which may be 2 years long, and having an interest in, maximizing your speed to patients to make a vaccine available, there could be a consideration to leverage an existing platform, to leverage a platform that may have received regulatory approval recently. And I think that, specific to the question posed regarding Merck's involvement, that is also another variable that received consideration and may have been an input into the choice or the decision, beyond which also the decision to have 2 vaccines in development. I would say that not every company is doing multiple vaccines. And I think that Merck has an interest in maximizing their opportunity for success, and they decided for themselves what is maximizing our opportunity for success looks like. It looks like having 2 programs. And finally, I think that whether you choose a viral vector, whether you choose a DNA-based vaccine, whether you choose a live attenuated virus vaccine, you might also have an interest in what does that dose look like to the patient. Is it a single dose vaccine? Is it a multi-dose vaccine? If it's a multi-dose vaccine, maybe you need 2 injections per patient. You just doubled the amount of drug substance that you need to produce. So if you're looking to vaccinate 3 billion people, now you need to make 6 billion doses compared to 3 billion. So that might also be a consideration, trying to have a long-term perspective towards making it available to the public.
All right, I have a question from Robert, and he asks, in situations like this where a vaccine might get emergency use authorization, do you ever worry about Resident Evil or I Am Legend situations where the virus might have even worse consequences than the vaccine might have even worse consequences than the original virus?
Take the question.
So thank you, MD. You finally let me go first. Maybe you want me to drown. That's a great question. I enjoy that. I appreciate that dialogue and that conversation. And I'm glad that we have an opportunity to touch upon this topic. And what I'd like to say or share towards that, maybe not specifically, give the answer that was being requested, but what I think is relevant here is to say that any company, Merck or any other company pursuing a vaccine for the public to end the pandemic is going to have a significant interest in making sure that they get it right, and making sure that if it does become available to patients, that it is safe and it is efficacious. Because previously in the world of either pharmaceuticals, if you're looking at small molecules, or biopharmaceuticals, I think both have examples of drugs or medications or vaccines that may have received regulatory approval, may have received FDA regulatory approval, and then after a certain amount of time on the market were later recalled. And from a biopharmaceutical company perspective, that's the worst possible outcome that could be achieved ever. A discard is better, discontinuing a program in clinical trials is better. The absolute worst thing to happen would be to need to recall a product, whether it's marketed as emergency use authorization or accelerated approval, or maybe it gets regulatory approval. Any company involved in vaccine development for COVID is going to have a significant interest, a major interest, in making sure that that's not something that has the opportunity to happen. And I think that regulatory agencies, either in the United States or around the world, who might have government pressure applied, might be, in a contentious discussion or a contentious argument there about whether or not it's appropriate to move forward. And, I think that the company that's pursuing that approval at that point is really going to be the additional voice to say, this isn't something that we're comfortable with, because if it were to need to be recalled, then we could be in a potential world of trouble. And nobody wants that for, patients' health. Nobody wants that from a business perspective of a company. So I think that there's a major significant interest to eliminate any possibility of that to happen.
I think Joe had a great response to that question. What I would like to add just a little bit about, if we're using emergency use authorization, I'm sure the regulatory agency knows what they're doing, and it will be a data-driven decision, not necessarily in a state where it's just like, okay, we're going to approve vaccine XYZ, right? The data needs to come in. It needs to show that the vaccine is actually working. It's actually safe for the patient. It's not causing any adverse effects. So, there would be a lot of consideration being taken before giving that emergency use authorization.
And just to finish MD's response, I think that whether you're making a decision in your personal life as a student, or making a decision as an international company to make a vaccine available to the public, you can only make the best decision you have with the information you have available when that decision is made. So if you're going through clinical trials and you're getting positive data back, that might be an appropriate time to say, we're going to move forward with emergency use authorization or accelerated approval. And then as additional data comes in, that might adjust the data that's available to say, this isn't something that's no longer appropriate. So I wouldn't say, if that were to happen, I wouldn't say that that would be a failure. I would just say that that's, making a data-driven decision. And if the data changes, then the decision could change.
So with respect to regulatory agencies, we have a question from Aaron, who has done some research and believes the mRNA vaccines being currently developed are the first being sent to FDA for approval. What sort of special challenges, benefits, et cetera, may this present to both production and approval within the FDA, if you're aware of these situations?
That's a good question. So I would say mRNA approach is very novel and it's very new. Although it's new, we do have siRNA therapeutics that have already been in the market, right? Taking the similar platform approaches, right? So there's a lot of lesson that could be learned from the success of those programs, but making this type of vaccine from the scratch, there would be, of course, a lot of challenges will be associated with it. I can't think of any specific one. I would say that, if you know a common platform that you have worked with before, it's much more easier to know how your overall process would look like, how are you going to go about your process development work? How are you going to go about choosing your facility to have the right set of equipment to do your job faster and the most robust way? Whereas if you're working with a new platform, it's very challenging to know what are some fermentation unit of operations you're going to be using. What are some purification unit of ops you're going to be using? That will be some challenges to be able to define those things and being able to make it in most robust way.
To add to MD's response, which was fantastic, I think that, with a different perspective or a perspective to a different area, if you have a vaccine platform that's previously been used and received successful regulatory approval with a complete data set for another disease, and you're leveraging the same platform, it might be easier. I don't know if easier is the right word. It might be something that can leverage existing data or previous data in support of pursuing emergency use or accelerated approval prior to the completion of phase 3 clinical trials. I think that if you're working with a vaccine construct that may not have been previously approved for use, that you might have additional challenges presented towards the accelerated approval outcome or the emergency use authorization outcome. And I think that those are different opportunities to balance, interest. And maybe, someone saying, well, we think that we can get into clinical trials more quickly, with approach A, and maybe someone else is saying, maybe we're going to be slower to clinical trials with approach B, but maybe we think approach B gives us a higher probability of receiving some form of emergency use or accelerated approval prior to completing clinical trials compared to, choice A.
Okay, I'm in a couple of questions together here for you. We have a question from Irene that asks, since antibodies don't seem to stay in the body long enough to maintain immunity to COVID after recovery, would it be possible for a future vaccine to be a one-time dose, or would boosters be necessary? And coupling that question with Jake's question, which is, he's heard or read that a COVID vaccine has a potential to be less effective and not last as long as other vaccines. Is this true? And if so, why?
You want to go first?
Yeah. Thank you again, MD. I love the deep end. So to respond to that, a great question. Thank you for bringing that forward. And I think that depending upon the type of virus that you're working with or the approach that you're taking, there may or may not be a stronger immune response. What is the threshold to generate an immune response, and how does maybe a viral vector meet that threshold compared to an RNA-based vaccine? Does it have twice as strong an immune response? How strong is that immune response? And how does it last over time? Great questions. I think like the tetanus shot, you're supposed to receive every 10 years. So maybe there's a general, example that could be relevant, specific to, COVID. My expectation would be that through the different programs going through development at different companies, my prediction would be that we're going to have multiple programs from multiple companies be successful at some point. And, maybe some of them are single dose, maybe some of them are multi-dose vaccines. And I think it's going to take, a significant length of time to definitively understand through a data-driven decision, as MD referenced, what does that look like moving forward. I don't think anyone can credibly sit here and say, well, if you get our vaccine today, then you're going to need another booster in 5 years. It's like, well, until you evaluate the behavior in a patient 5 years from now, I don't see how you can definitively make that claim. So I think that, the more immediate interest is to demonstrate that the vaccine generates an immune response, is safe for patients, and is something that is appropriate through regulatory scrutiny to make available to the public. And then after that point, moving forward with a longer term perspective towards the future, say, is this a vaccine that's been approved that requires a booster? Do patients still show, antibodies X amount of time later? And again, make the driven decision.
I think that's a great response, Joe. The only thing I would like to add to that is what we need to understand here is that vaccine had been around for a long, long time now, and we did not ever see a need for developing vaccine in such a short time. Like usually vaccine development takes a lot of time, and companies do have a lot of time to go through clinical trials and find out the most effective way they're going to have their approach in vaccines. But here, due to the tight timeline, I would say the scientists and the engineers in different companies are doing such a great work for it to come, and we would not know entirely until the time comes and we get to see more data. COVID is not the only RNA-based virus, right? Like in past, we have seen Ebola, we have seen polio, we have seen influenza, right? And we were able to develop effective vaccines for those products. So, I believe with time, we would be able to come up with solution, to tackle this challenge.
All right, I have a quick follow-up question from Dr. Russell, and they ask, would you take a vaccine that's been released before completing phase 3?
My personal answer would be no. I would want it to go through phase 3 clinical trial and see the full data before taking the vaccine. I don't know, Joe, if you would be willing to take the vaccine.
I think that's a personal question that's going to depend upon different levels of interest that individuals might have in being part of something that's new. I think that there are a lot of folks who are very brave to volunteer and offer consent to be part of the clinical trial process. So, some people might feel comfortable being involved in the clinical trial process. Some folks might feel comfortable receiving a vaccine that's classified under emergency use authorization or accelerated approval. And some people might only be looking at a regulatory approved vaccine, and maybe they're only going to be considering it, 5 or 10 years after it's received regulatory approval, and there's, even more information and data available about how it's tolerated in patients. For me specifically, my hometown is very close to New York City. I've spent a lot of time there over the last roughly 8 months that we've all been, enduring this unique global challenge, if you will. And, in that area of the country, in the United States, there have been a number of cases, at a time, maybe back in May or June, I'd say we were probably more elevated compared to other areas of the country or the world. And, I'm sitting here tonight with you presenting, and it's like, do I have antibodies and was I asymptomatic? I can't definitively prove that. But, what level of risk do I feel comfortable with? How mindful am I being? What do I think my behaviors are doing to present an opportunity for me to be infected? And how do I balance that against an opportunity to be vaccinated? So, to answer the question specifically, I'm going to lean towards saying no. However, I think that it varies on a case-by-case basis. And if I was an intensive care unit nurse who was in a major hospital caring for patients on a daily basis, that might influence my decision.
All right, so I have a question from Ethan, and they ask, are there any manufacturing quirks due to COVID-19 being an RNA-based virus?
So I would say that, as I said in my previous response, that COVID is not the only RNA-based virus. We have had, previously developed vaccines that were RNA-based and, had success with that. So I'm sure that COVID specifically will have its own challenges, but, choosing a platform and overall, developing a platform, I wouldn't think that it will be much different than other RNA-based vaccines that we have seen.
So again, building upon MD's robust response, I think that basis of the virus presents an opportunity, and this is my opinion, presents an opportunity to influence the behavior you might see in different cell lines, which might then affect the data-driven decision you might make to say, I'm going with E. coli, I'm going with mammalian cells, I'm going with a type of yeast cell because I think it's most optimal. And if you're not working with, an RNA-based virus, maybe that is an opportunity to influence that decision at that time as you're going through development in the product life cycle.
So we have a question from Matthias here. Thinking of time of construction, new plants, retrofitting, market availability, how do you see the role of single-use technologies in dealing with this pandemic and the future of bioprocessing and engineering?
That's a great question. So I would say that single-use technology is the future. That's my opinion, by the way. I mean, I don't know how other experts will feel. I don't know how Joe will feel. But the advantage with single-use technology, right? Like, you can develop facilities. Now, these days, when we develop facilities, right, we have to keep in mind of, what kind of tank we're going to put in, what kind of cleaning procedure we're going to develop for it. And then there's also cleaning validation that we have to do every year. Then if it's a sterilizing tank, that we have to do sterilization or steam in place validation every year as well. Those are very time consuming. And every time cleaning a tank or cleaning an equipment could take 4, 5, 6 hours, depending on how complex the tank is and how much cleaning needs to be taken into consideration. So single-use technology would be the solution, where it would be very fast, where you can design your facility such a way. And let's say if a vaccine or if a biologics didn't work out, you can put another vaccine or biologics in the similar settings where you don't need to worry about moving a tank or fitting another tank. And you would be able to save significant amount of time after processing every time, not worrying about cleaning, not worrying about exceeding dirty hole time, which is bad, by the way. If you ever exceed the dirty hole time, which means the maximum time you can keep a tank or equipment dirty, if you exceed that, then you have to do a cleaning verification, which will require submitting samples to the lab, and the lab result needs to get back before proceeding with your manufacturing activities. So with single-use technologies, all these bottleneck could be reduced, and I believe that manufacturing could be more robust.
So I love the question. It's something that I've been looking at for the last 20 minutes, and I'm very excited that we finally had the opportunity to address it. So thank you, Matthias, for the question. MD, I really enjoy your response, and to build upon that from a research and development perspective, and speak specifically towards new plant construction and retrofitting. I'll start with the R&D portion and then move specifically to those manufacturing items. But in research and development, you're not going to have an interest, or you're not going to have the resources or time available to go through cleaning validation studies to validate your process. You're not going to be spending that much time. You're not going to be making 80 batches a year. It could be 3, 4, 5, 6, 7, 8 batches in the entire research and development space. So you're not going to be interested in pursuing cleaning validation. So you're going to be working with single-use technologies, and that's going to allow you to quickly go from one clinical program to another clinical program, either in process development or in clinical supply manufacturing environment. In addition to that, in the clinical space, you're also going to be promoting a modular environment where you can say, we're going to go from this tank to this filtration system, or that filtration system to this bioreactor, and you can quickly adjust that compared to a hard piped plant where you're not going to have the same level of flexibility. Moving over and speaking specifically towards retrofitting or the construction of a new building, at the end of the day, when you start looking at how quickly you can make a product available to the public, your two main considerations is like, in this hand I have time, and in this hand I have money. And the more money I'm willing to spend, the faster things happen. And while things happen more quickly, you want to make sure you're not compromising the quality of your building, your unit operations, anything involved. So the more money you spend, the faster you can make things happen. And I think that's what we're really seeing here with vaccine development specific to the pandemic, how our companies in phase 3 clinical trials, less than 12 months after the virus was discovered, Well, if you make a big enough investment, you can get in clinical trials as quickly as you want. Unfortunately, because you're going through all the process development phases in parallel, you're buying equipment, you're buying filters, you're buying everything you need on risk in parallel. And then if you end up not using it, you don't really care because you decide to spend a whole bunch of money that you had authorization to move forward with. Specific to a new building with single use, you're eliminating a utility need, you're eliminating a utility load, and you're minimizing the amount of equipment that needs to be installed. You could have an entire floor of a building dedicated to clean steam or dedicated to pharmaceutical grade water supply, which could be at a variety of different temperatures or pressures regulated throughout your process. So with single-use technology, you're eliminating all of that. I think you're maximizing your opportunity to achieve the highest exit cycle, or excuse me, the lowest exit cycle time possible to achieve the highest amount of product produced possible to get to patients. And I think at some point, you're going to see a financial trade-off where if I make 50 batches a year, single-use technology is more affordable than maybe using utilities for clean in place and steam in place and cleaning verification and all the resources and employees that would require. And maybe if you're going to 100 batches a year, the maximum output of the facility achievable with single-use technology, maybe you're not having the most optimal financial performance, but if your primary interest is in getting to the market and making it available and being one of the first 1, 2, 3, 4 companies to do so and start ending the pandemic, I think that's a trade-off that's going to be willing to happen. So I think that single-use technology is going to play a massive role in the ending the pandemic and the future of bioprocess engineering. And thank you again for the question. Fantastic question.
All right, we have a question from Wei, and they ask, what parts of the vaccine development / pharmaceutical industry do chemical engineers typically work with?
That's a good question. So I would say chemical engineer can work in variety of these developmental activities, right? Of course, not in so much in basic research, unless someone has like very specific knowledge about or research experience about the basic research, but they could work anywhere in clinical manufacturing. They could be involved in process development, process characterization, process validation, involving in large-scale manufacturing on day-to-day support as a process engineers. So they could contribute in many different ways, whether it's in the laboratory, helping to scale up process, or in the manufacturing settings, ensuring that we're making the same quality of product every time. They can have a significant impact in both of those areas.
So again, I enjoy Angie's response, and thank you for the question. I think that chemical engineering is one of the majors that maximizes the opportunity that becomes available to graduates who are looking to be involved in biopharmaceuticals. You have a variety of different doors that are open to you. You can go into large molecules, which are vaccines and biologics. You can go into small molecules, which are pills and tablets. And that's like the first decision. After that, you could be involved in manufacturing. If you're talking about large molecules specific to vaccine development, you could be in fermentation, you could be in purification, you could be in formulation, you could be in filling, you could be in packaging, you could be in supply chain, and you could be in quality, you could be in operations, you could be in technical operations. There are a number of different opportunities for you to pursue, and that's all on the manufacturing side. Transitioning over to the research side, you can be in process development for upstream and downstream unit operations. You can be in process development for large molecule or small molecule. You can be involved in a pilot plant. So I think chemical engineering is a very broad educational opportunity to maximize how competitive you are to pursue one of those opportunities, where another degree that might not be chemical engineering, maybe it's another type of engineering, maybe it's a type of science, might not make all of those choices available.
I'm going to ask a question that I've been thinking about since I heard MD talk about supply chain early. How much of a concern is supply chain in the development of these vaccines? It didn't occur to me until I heard this mentioned. Is there a real risk of vaccine development being delayed due to not being able to get, your separations equipment or something in?
So Dr. Mancucci, thank you for the question. I'll say, speaking from what my perspective is, which might be unique, being involved in Merck research labs and being involved in vaccine programs and being involved in COVID programs at Merck, I think that the supply chain might be the rate limiting factor in this equation as we progress towards success. If we're working with a single-use bioreactor and we need that for our fermentation or our upstream process, and the vendor for the single-use bioreactor bag says we can't get that to you until December, well, we're not doing anything until December. So then it's like, okay, well, maybe we're going to go to an alternative vendor. And maybe they say, well, you have to wait until February of next year. And then maybe we, escalate that. And then we have, somebody at the executive level say, well, here's a whole bunch of money. Can we get that next week? So there are different conversations that happen at different levels of the organization to maximize the opportunity for success related to supply chain oriented issues specific to development. And I think that, when you start incorporating single-use technologies and moving away from stainless steel or, a cleaning verification, approach, you invite more opportunity for supply chain to become a focus area. And I think that's something that, you need to evaluate, and each company would prioritize based upon their own, decision-making process. But I think at the end of the day, considering the unfortunate nature of the pandemic, there's going to be an interest in spending a lot of money to get things to happen very quickly and to eliminate any supply chain related accessible hurdles.
I think that it's a very good question, Dr. Minicucci. I would say, like similar thing as Joe had said, right? Like it's very important to choose the right set of vendor. It's not always about who can give me this equipment as the most lower cost, but who can also provide me the best service possible. So when the equipment is not working and I need help with troubleshooting, they could also give me the expert at the quickest time so I could get my equipment running and start doing whatever I was intended to do earlier. And also making sure that the quality of the product that they're giving us every time, it's at the best quality, because we are with their raw material supply or whatever we take from vendor, if it's not the best quality, then it will impact how our process will behave. So, that's why it's always important to choose the right vendor and have good vendor relationship to be ahead.
All right, we have a question kind of in the same topic. They ask, bioprocessing has historically been based on batch processes, and now there's a move towards continuous processes. Could you comment on this change and the implications on the future?
So again, thank you for the question. I really appreciate that. I see it's related to the origin of the single-use question, which I really enjoyed. So very excited. So thank you. And I think to answer that, I think there are opportunities to move towards continuous processing, but I think that you might always see a need for batch processing. And maybe you're going to be minimizing your risk with overlap and increasing the opportunity for batch overlap to really drive down your exit cycle time. A specific example that comes to mind as an opportunity for a continuous process might be to fluidize a bioreactor. Maybe you have a continuous feed of media that's a defined flow rate to keep your cells suspended in the middle of the reactor while you're also carrying out your product maybe through the top. So that allows your cells to continue to live there and exist and produce your protein of interest, but you're continuously capturing your product. So I think that might be an example of an area that might be feasible for continuous processing. If you were to look at, maybe a vaccine that has multiple serotypes involved, I really think it's going to be a big challenge to employ continuous processing there. But I think that there's definitely an interest and a fit for it in the right place, in the right unit operation.
Yeah, I think that's a very good response. I don't have anything to add.
We're winding down in terms of time. We have one more quick question, and then I'd like to wrap up. Dr. Russell asks if either of you have read Crisis in the Red Zone about the Ebola vaccine development, and what is being used, if you have read it, what's being used from that knowledge, and what new developments have come about since?
That's a good question, Dr. Russell. I have not read the book. I have heard of it, though. It's on my do list, things to do. So I won't be as effective answering this question, but I'm sure since then, you know, we did have a lot of process improvement or process learning in the vaccine manufacturing area that was implemented, which the success could be shown in the case of COVID, that even with the tighter timeline, right, like, you know, Pfizer, AstraZeneca, J&J, they're going to have their phase 3 clinical trial concluded in 2 years, which is very significant because, like, you know, a typical vaccine life cycle is, you know, usually very extended, and you know that 2 years is a very, very accelerated timeline.
So thank you for the question, and MD, thank you for your thoughts. Like MD, I have not read the book, but I'm interested to speak towards the Ebola vaccine development component of the question. And, you know, having Merck successfully receive FDA approval for the Ebola vaccine and being involved in research and development and looking at public statements that have been made about Merck's involvement in COVID development, you know, leveraging the Ebola experience that they have is something that, in my opinion, is valuable and an influence upon, you know, program-based decisions or construct-based decisions about how to approach and move forward with development. I think that, you know, if Merck is thinking about internally commercializing one of their vaccine candidates, you know, if they were to decide to do that instead of moving towards a CMO, then that's something that might be valuable to say, you know, maybe this is a similar type of virus, or maybe this is a similar type of cell line that we might have a lot of comfort or experience with, and that could have a positive influence. However, I think every time you go through the development process, as long as you have a broad perspective, you'll always learn something new. So I think it's an opportunity to say, well, we learned from, you know, working with Ebola, and now we're going to learn from working with COVID. And I think a big influence in the learnings from the COVID experience is going to be the speed component. What happens when you're going through phase 1, and then, you know, less than 6 months later, you're going through phase 3, and, you know, what does that look like? How does that influence your choices, your decisions, your involvement of commercialization? You know, how early should you get commercialization involved? Should it be involved before phase 1? You know, how confident are you in the program? So I think that hindsight is always 20/20, and it's a lot easier to sit there in, you know, 2022 or 2023 and say, this is what a better approach would have been, or, you know, maybe an mRNA vaccine is definitely going to be successful, or a viral vector vaccine is definitely going to be successful. But, you know, again, it goes back to the idea you can only make the best decision you have with the information you have when you make that decision. And, you know, I think having a holistic approach and incorporating as many perspectives and, you know, the most robust data sets as possible and that are available is going to be the primary driver towards success in that area, either leveraging Ebola experience or other companies leveraging previous vaccine development experience.
So I want to take the opportunity now, we're running out of time this evening. I want to take the opportunity to speak from a personal point of view. I had the opportunity to teach both Joe and MD previously, and I want to highlight that they share a common characteristic in that they spent their education, at least the time that I saw it, full of questions. They asked so many questions. They asked questions of everyone, and they pursued knowledge with an energy that really provided a lot of passion. And it provided a lot of I would say it's a model method to be a student. And I'd say that anyway, even if you weren't doing this tonight. And one of the things that from me, what I want to say is that I think of the effort that you are putting forth in vaccine development right now as one of the most heroic examples of engineering and science that the world has ever seen. This to me is the space race, getting somebody on the moon, but with the world suffering under a global pandemic. And having had you both as students, I couldn't ask for any two better students to be there doing it on a daily basis. It means a lot to me. to know that one of the vaccines or two of the vaccines and all of the vaccines you're working on are in your hands. And so on behalf of everyone here today, on behalf of all of us as a general public, I want to extend our deepest thank you to you, not just for the presentation tonight, but also for all of your efforts in trying to deliver a safe and effective vaccine to us as quickly as possible. It really is an astonishing effort, and thank you so much.
So, Dr. Minicucci, it was a privilege when I was a first-year student back in, I believe it's 2015, and 5 years later, it's still a privilege. And for you to have your opinion, grow and influenced in such a significant way, in such a positive way, towards both myself and MD, we're very, proud of that. We're grateful for that, and it resonates with us, and we're glad that we set a positive example moving forward, and we appreciate your invitation this evening to be hosted by Montana State, even though we're in Pennsylvania right now, and contribute to this discussion, contribute to this educational opportunity to provide insight, to provide a real world life experience perspective to help folks not only in America but around the world who are able to join us have more information, be more educated, not only for themselves but for their families, and moving forward, hopefully this is an experience that we'll all be putting behind us sooner rather than later. And in addition to that, maybe some folks on the call will be inspired in their future educational or career choices as well. Maybe they're going to be saying, I'd love to be involved in biopharmaceuticals, or maybe I'm saying, wow, I'm really able to identify a passion in another area. And I think at the end of the day, as long as you're passionate about what you do, that's really going to maximize your level of engagement, which is going to promote success and advancement, and more importantly than all of that, fulfillment. And I think that that's something that MD and I sitting here tonight can definitely say, we're very fortunate to be the beneficiary of, significant, severe fulfillment in the best way possible, mainly thanks to not only your guidance when we were undergraduates, but also the continued professional relationship and the opportunity to be here with you all tonight. So thank you very much.
I think Joe said it perfectly. I'll just like to add that it was a great privilege to be here presenting. And Dr. Minicucci, you have been a very special figure during our undergraduate time, and we still continue to be in touch. I mean, it's we have learned a lot from you during our undergraduate time, and you did find a way to motivate us even more, although we were motivated already, and pushed us to be better. That's something that we carry on our daily base. And thank you to everyone for joining us and for their time. Really enjoyed the discussion and the question that came along, and it was definitely a good experience.
All right, everybody, thank you. Austin, if you can post our links again. And Joe and MD, people are left wanting more. So they're asking how they can get in touch with you or hear more from you. I'm going to ask you, if you're interested in that, to contact us at MSU. Go to msuaiche.org and contact us, tweet using our hashtag or our whatever the kids say these days, and promote it, and then we'll try to put you in touch. And if we have future events, make sure you fill out the event specific form so that we can inform you about the next time we have a presentation. Again, Autumn, I wanted to point out, Autumn said thank you. A number of people have said thank you for your efforts. Thank you very much, gentlemen. Thank you for your time. Thank you everyone for attending, and good night.